Good morning everyone. I am Harita, DL Botany at Telangana Tribal Welfare Residential Degree College for Men, Sangareddy. Today I am going to deal a topic from plant physiology. We'll begin with plant physiology because it is one of the major topics when students are aiming for their PG entrance exams. So you can see in the first slide different growth phases of the plant from a seed to till the abscission. What do you observe? What do you understand by these pictures? When you look into these pictures, you see that you can see seeds germinating into a small seedling, later growing into a small plant, and the plant after a certain period of vegetative growth initiates the flowering, fruiting, and after a period of time, as the old plant parts decay, it leads to what is known as senescence. What do you think is triggering all these structures then? What is this physiological process is known as? This is nothing but, dear friends, it is growth and development. Whenever we talk about growth and development, it is one of the major complex physiological processes in any living organism. Here we are dealing with plants, so we'll discuss growth and development patterns in the plant. So what do we observe? What is growth? Remember that growth is a measurable process which can be measured. That is, if a plant is growing, it would show how it grows by increase in the size of its height, weight and girth. But if we are looking at development, development is not a measurable quantity, but it is definitely associated with the growth. So what does development mean? Development means differentiation. Differentiation of a particular cell into a proper tissue and its functioning. So cellular differentiation, which will further lead to what is known as morphogenesis, is called as differentiation. Okay, so these both are very important interrelated processes when we are talking about the growth. Okay, so growth and development may be abrupt or it may be gradual in nature. So if we take a plant into consideration, there are three major parts of life cycle. That is, first one is germination, second one is flowering, third one is senescence. If we are talking about growth in particularly, now growth of a plant can be divisible into two parts, that is vegetative phase of its life and reproductive phase of its life. Vegetative phase of life starts from seedling stage and till the flower initiation starts, and the reproductive stage is triggered from flower initiation till the development of the fruit and seed setting. And we also see that the growth of the plant is once again divisible into determinate growth and the indeterminate growth. What do you mean by determinate growth and indeterminate growth? We find that if you look into a branch, you find that there would be specific number of leaves. If there is an inflorescence, you come across a specific group of flowers. So a determinate growth is characterized by production of certain number. So where we see this determinate growth in a plant, especially in development of fruits, flowers and leaves. Then there are places where the growth is indeterminate in nature, that is, it continuous. Where are those points? That is the shoot apex and the root apex. Okay. Now when we have come to root apex and shoot apex, let's more elaborate on this in the introductory topic. So when we are talking about what are those zones, what are those places where this growth pattern occurs, then we find that there are certain places, localized portions in a plant where meristematic activity takes place. That is, meristems are present in that particular place which triggers the growth of that particular area or plant part. Okay, so depending upon its position, function, we know about the classification of the meristems. So we will come across primary meristems which are there from the embryonic stage. Then we have secondary meristems which help in secondary growth, that is in cortex and vascular region. And if you are taking grasses as example, we find that the growth is mostly intercalary in nature. So these are the few things that are associated with the growth. Let me add two more points when we are talking about growth. That is, what are the factors that are responsible for the growth? If you are talking about those processes, there are definitely the abiotic factors. They are the major influence on how a plant should grow, how much it should grow. What are those abiotic factors? The first one is light, second one is interrelated factor called as temperature, water and nutritional requirements. Okay, so but those abiotic factors may vary from one plant group to other plant group depending upon in which region this particular plant grows. If we are looking into the light, one of the major abiotic factor, the intensity, duration, the type, kind of light is very important for that particular plant growth. And in visible spectrum, we observe that red and the far red light are the most preferred for plant growth. And if we are looking at the temperature variation is quite diverse depending upon the kind of plants. If we are looking into Arctic plants, 10 degrees is the optimum temperature for its growth. If we are looking into temperate plants, it is 10 to 25 degrees or 15 to 20 degrees, and in tropical plants it varies from 25 to 30 degrees also. And if you are talking about water, we know that water is one of the most physiologically important abiotic factor without which we cannot imagine life on earth. And of course, depending upon the kind of plant, their physiological aspect varies. Now, if we are looking at the last factor, that is the nutritional factors, we know that each plant requires a minimum of 16 inorganic elements which are very essential for the plant growth. And if we are going to characterize them, if we want to see how healthy a plant is, we have to take into consideration C/N ratio, that is carbohydrate by nitrogenous matter ratio. If the C/N ratio is low, we will observe that the plant grows more vegetatively. If the C/N ratio is more, then we find that there is less amount of physical growth and more reproductive growth we can see. Okay. So, these are all about the abiotic factors. Then there should be some triggering factor inside the plant also. Of course, that lies in DNA, RNA. Without them, if there are no proteins to growth factor which will trigger growth, the plant growth cannot happen. So, we are going to discuss the triggering factors that are present inside the cell which will lead to growth and development of the plant. It was not today. In 18th century itself, Julius von Sachs was the first person who has put forward an idea that there are certain kind of chemical substances in the plant that would enhance the growth of a particular plant. So, the foundation for the study of plant hormones was laid there over there itself. So, today our topic is plant hormones or growth bioregulators. Let's move on with our slides then, friends. Now, when I am going to deal with this topic, what we are going to precisely know what is each kind of a phytohormone, what is its definition, what are its characteristic features, what is its outermost classification, and what are its functions. Because without them, we cannot imagine a plant to grow in its size or weight or complete its phenological life cycle. So, look at the slide now. There are two more questions over here. What are plant hormones? What are the different kinds of hormones and what effect does they have on the plants? These are few questions on which our topic is going to run today. So, let us begin with this one. So, if we are talking about plant hormones, these plant hormones are organic substances which are produced in very little quantity. There are specifications for it also, like they are produced in very less concentrations, they are produced at one point, but the trigger point or where they exhibit their nature is at some other place. Okay. So, for a long period of time, plant hormones have been are used for defining these kind of chemical substances, but as the knowledge about them developed, hormones are more associated with the animals rather than plants, because their physiology is completely different from the plants. So, now the most appropriate word that we are going to use is plant growth bioregulators or the growth substances. Hope it is clear. Now, let us move on to the next one. So, what are the characteristic features of the growth substances? These are the few of them. They are endogenously produced, that is produced within the plant and in very less concentration, very small concentration in nanograms, micrograms they are produced, but they can have a very major influence. And since I told you that they are produced at one part and show their effect on the other plant part, it means that they have to travel, they have to transport, and that is because the nature of the growth substances itself is they are highly diffusible. They can move from one part of the plant to the other through various modes. And do you know what is the most popular mode? That is the xylem and the phloem. Of course, few parenchyma cells in the cortex also, they can move through that. And we have to remember that each plant hormone has its own specific function. So, there is no overlapping over here. And plant hormones, when we are talking about plant hormones or growth substances, they can have two opposite effects also. Certain plant growth substances have as promoting substances, few may be inhibiting the growth. Why? We will deal with it when we progress further. Okay. Now, let us look at the classification of the growth substances over here. We can classify them into auxins, gibberellins, abscisic acid, ethylene and brasinosteroids. Of course, auxins is not cannot be seen on the screen right now. So, the primary growth substance is auxin, followed by gibberellins, abscisic acid, ethylene and the newly identified plant hormones, that is brasinosteroids. Besides that also, we come across more different kinds of plant hormones naturally produced within the plant, we have comaric acid, cinnamic acid, ferulic acid, jasmonic acid. These are all also produced by the plants, and it is not only all kinds of plants cannot produce all kind of hormones. So, they may be present in specific group of plants depending upon the kind of species. Okay. So, we have just seen how we could have classified the plant hormones, but depending upon their effect, once again we can classify them into the promoters and the inhibitors. So, among the promoters, growth promoting plant regulators are auxins, cytokinins, gibberellins and brasinosteroids, whereas inhibitors are primarily ethylene and abscisic acid. So, these are the chemical structures of all the plant hormones, few of them of course among them. So, you can see that they are almost are ringed structures, phenolic compounds, aromatic compounds, and now let us look into each of them. Now, when I deal with the each of the phytohormone, we will focus on how it was isolated, what is its structure, what are its physiological aspects and what is its mode of action. Now, first we will begin with the auxins. We know that the auxins, before Went has discovered it, they were discovered in the as callus structures in the lab chemical analysis in human urine, rhizopus, etc. So, finally, F.W. Went was able to isolate that auxin from the avena, wheat and maize. And if we are looking at the definition of the auxin, Timman has rightly put that these are organic substances which help in promoting the growth of the plant. So, promoting the growth of the plant can be various. It can be cell growth, it can be a tissue growth, or it can be any other phenological development over here. And if we are looking at the occurrence, auxins are found in all group of plants, that is from the lower group algae to the angiosperms, we can find them growing present everywhere in every group of plant. Not only that, if we are looking into the plant itself, these can be found in all parts of the plant. But basically, wherever there are root tips and shoot tips, in those places the concentration of the auxin production is more compared to the other one. So, in the cytosol of the cell, these auxins are produced. And how do they move? I said that most of all the hormones are diffusible, they need to move. Yes, their movement is only in one direction, that is the polar direction, and they traverse through the phloem and parenchyma, but for their movement they need energy, that is by active transport they can move from one place to the other place. So, the movement, you remember that usually the auxins are produced more on the root tips and the shoot tips. So, if the auxin is produced in the shoot tip, it has to traverse down to show its effect on the particular plant part. Okay. So, you can see the chemical nature of the auxins also. They are mostly indole compounds. Indole compounds are all aromatic carbon compounds, and besides IAA, that is the indole acetic acid, which was the first identified auxin, we have many other indole compounds that are present in the plant, which are converted into IAA by different kinds of enzymes. Okay. Not only that, we find that each auxin will have an indole ring, a carboxyl group, and at least one carbon atom will be present between the ring and the carboxyl group. Okay. Now, let us go further. So, we find that the auxins are usually that are produced endogenously naturally, but looking at the major effects that they show on the plant, they are majorly used in horticulture, floriculture practices, and of course in agriculture. So, there are many other synthetic auxins are there that are that have come into the horticulture market, where using the same natural chemical structure, artificial auxins are produced and are promoted to increase the yield in agriculture or the related crops. So you can see the natural different kinds of auxins that are present, that is indole acetaldehyde, indole ethanol, etc. And the synthetic auxins, 2,4-D is quite famous. You might be knowing it's a weak weedicide, and 2,4,5-trichloro, 2,4,5-T is also quite popular among the herbicides, weak herbicides. Okay. So in most of them, these auxins are used in tissue culture labs for root promotion, growing of the root promotion and other horticulture practices. Now let's go further now. Now let's try to understand the physiological effects of the auxins now. Okay. So what is the primary effect of the auxins? We come across cell enlargement, cell division, apical cell dominance and the lateral buds. Okay. So if we are looking at the cell enlargement, this cell enlargement and cell division are quite interrelated. Now the auxins promote cell division, especially in the cambial region. So they give rise to secondary xylem and secondary phloem. Not only that, the auxins have a very prominent role in maintaining the apical dominance. Now what do you mean by apical dominance? Apical dominance is nothing but where the apical bud being active, because the auxins are synthesized in the tip regions, we find that the lateral buds that are produced beneath the apical buds always are in inhibition stage. So what do we need to do? Till the auxins are present in concentration, lateral buds cannot proliferate or grow. So the auxins help in maintaining the apical dominance in plants. And as I told you, they trigger root formation. So in horticulture practices, what is the common practice is that they take small pieces of stem, cut off the apical portion, clean off all the leaves and put them in a soil, and after a few days you find adventitious roots growing beneath them. Okay. Not only that, they also trigger tropism. Tropism are plant movements. So they favor both phototropism and geotropism. Remember one point, that auxins usually are produced in more concentration in dark. So usually you might have observed in balconies, the plants that are placed in the balconies drooping outside the balcony. Have you ever understood why that particular plant is drooping, whereas a normal plant which is there in a garden doesn't droop? This is only because of differential light that is falling on the balcony. So auxins are produced more on the darker side compared to the lighter side, so they lead to more production, hence the stem bends towards the light. So they favor tropism, that is in the root they force the roots to go down, deep down into the soil, whereas outside aerially they promote phototropism. Okay. Now looking at flower formation, we find that in pineapples these particular auxins promote plant formation, flower formation, and not only that, they also induce parthenocarpy. What is parthenocarpy? Parthenocarpy is induction fruit setting without sexual reproduction. So in few plants like tomatoes, in pears, and in such kind of plants, they do promote parthenocarpy. Yes, and abscission of leaves and fruit, they inhibit the abscission. What is abscission? Abscission is falling off of the plant parts after they attain certain amount of maturity. So what happens? How does the abscission occur? There is a small weak layer that is formed, which we call it as abscission layer, between the plant part that has to be separated from the main plant. As these abscission layer cells are quite weak in nature, after a certain period of time, that particular plant parts separates from the actual plant and falls off. So in most of the cases, we find that auxins, when they are present, they inhibit abscission. And not only that, as I told you, auxins are majorly used in horticulture practices. When they are used mildly, exogenously, they can help in weed control, especially 2,4-D we have seen. And not only that, they have an interesting phenomena also, like in cucurbitaceae we know that male flowers and female flowers are produced separately, that is we have male plant and female plant. They help in auxins, if they are treated with auxins, they help in change in sex expression, that is instead of male flowers on a male plant, female flowers are produced. These are the few physiological effects of the auxins. Let us move further. These pictures depict you whatever we have studied now, that is root promoting nature, new apical bud formation and the other things. Now let us discuss about the mode of action, how this particular auxins are going to affect what. you can see that almost all plant hormones have a bearing on the DNA and RNA synthesis machinery. They have either directly or indirectly, they keep on acting or interfering or promoting DNA synthesis or RNA synthesis. And these auxin have a receptor complex where they are fused with certain specific proteins which help in RNA synthesis. And not only that, as I told you, in case of cell enlargement or cell, we find that these particular auxins, when they are present, they help in dissolution of middle lamella. This is how their variation, their mode of action varies depending upon their nature. Okay. Now, look into the agriculture and horticulture applications of the auxins. We have just discussed them priorly also in the previous slide. You can just go through them, that they help in vegetative propagation, they help, they prevent the sprouting of the bud, so that the storage life of that particular plant that we might be transporting, or an agriculture practitioner wants to retain it, can be done using that. They also help in preventing leaf fall, they help in development of parthenocarpic fruit, help in fruit setting, and also prevent the premature fruit drop. Now, this was about the first kind of growth promoting substance, that is the auxins. Now, we will quickly move to the next interesting growth substance, that are the gibberellins. Now, these gibberellins, the main source of these gibberellins is from the fungus known as Gibberella fujikuroi. This particular fungus used to cause what is known as foolish seedling disease in rice plants. So, it was Kurosawa who has identified and took it out, but Yakuba and Hayashi were the first ones who have recognized that gibberellins from this particular plant. So, what does this gibberellins do? They also promote growth, but what kind of growth? They help in growing in the height of the plant. So, they help in stem elongation because of cell enlargement. Okay. And as we have seen in the auxins, these are also found in all parts of the plant, but the main concentration of the gibberellins is in the seeds and in the young plant parts, that is in the young leaves. Okay. Till now, 50 different kinds of gibberellins has been isolated from different plants, and we refer it with GA subscript 1 to 50 like that. So, except G2, G1 to G9 are found in plants, and G2, that is gibberellin 2, is found only in the fungus. And if we are talking about the movement, these gibberellins are non-polar in movement. They can traverse in both the directions, and they are quite passive in their movement. They do not need any kind of energy for their movement, and usually they pass through the vascular tissues, that is the xylem and the phloem. If you are looking at the chemical structure, you can see that the gibberellins are basically 4 or 5 ringed diterpenoids, and how they are synthesized using the mevalonic pathway. This mevalonic pathway occurs in the plastids. So, along with the terpenes, sterols and terpenoids, even these gibberellins are synthesized using that pathway. Now, we will quickly look into the physiological effects of the gibberellins. Now, as I told you in the introduction itself, these gibberellins promote stem elongation. So, in few kinds of dwarf mutants, if you are taking an example of maize plant or certain kind of peas where there are dwarf varieties, they are dwarf because they cannot synthesize gibberellins inside themselves. So, exogenous application of these gibberellins can help in elongation of the stem and coming back to the natural form. Not only that, if you are looking into plants like radish or cabbage, we find that they promote bolting and flowering. Now, what is this bolting? Bolting is the elongation of the stem from a rosette. Okay. Now, this bolting has an interconnection. Only when that bolting occurs, flowering can occur in that particular plant. So, these two are interrelated factors which whose effect is caused by the gibberellins. Now, the third physiological effect it can show is in certain kind of plants like long day plants, we find that they can substitute cold treatment. Actually, cold treatment is required to break seed dormancy or bud dormancy in certain cases. So, we find that mostly in biennials, certain kinds of biennials and certain kinds of long day plants, we instead of that cold treatment, if the gibberellins are used, this can substitute the cold treatment. Now, breaking of dormancy is also one other important factor. Now we know that embryo derives its energy from the endosperm, and endosperm stores basically starch and proteins. Now these starch and proteins are not easily available to the plant. It has to be broken down using the enzymes. What kind of enzymes? Amylases and proteases. Now these gibberellins that are present in the seed will trigger the formation of these particular enzymes, which act upon the endosperm, releasing the required nutrition to the growing embryo till it comes out in the form of a small plantlet. Not only that, in certain kind of plants, fruit plants especially like apple, peaches and pears, we find that it can induce parthenocarpy. Once again, it can be explored for horticulture practices. Then not only that, gibberellins, as they has the affinity to increase the size, we find that in certain kind of plants like we have camellias, we have dandelions, like ornamental plants, if gibberellins are applied exogenously, they can promote increase in the size of their morphological parts like leaves, flowers and fruit. And of course, seed germination, we have just talked about it, yes, and once again they also may lead to flowering and sex expression. In certain plants, they also help in differential growth. So you can see in this picture, bolting on one side, grape set movement, the size of the grapes are quite small on one side. After their gibberellin application, they have increased, the fruit set has increased. They help in germination, they help in stem elongation, and also papaya plant, you can see where in the first plant there were no flowers, but after an external application of gibberellins, the flowers have been produced. Okay. Now let us look into the mode of action. The primary action of the gibberellins is stem elongation and cell elongation. We have already discussed about it, isn't it or not? Yes, once again they show their preference that they do interfere in RNA and protein synthesis also. So they aid in RNA and DNA protein synthesis. One more thing that is most important is gibberellins have a control triggering activity on the mitotic cycle too. Yes, so they too can bind with specific proteins and exhibit collateral effect. So these are the different modes of actions of the gibberellins. So let us move on to the commercial uses of the gibberellins. As we have discussed in the physiological effects, the same things we can see one more time, like on horticulture plants, like in floriculture, for increasing the grape cluster, these gibberellins are popularly used, and for production of seedless grapes, that is parthenocarpy, yes, seedless grape, they can enhance alpha amylase activity, once again, which can be used in beer industry, and they increase the content of sugar in the sugar cane. So these are the few things which can be employed very successfully to get maximum yield and profit using the gibberellins artificially, exogenously. Now we will move to the next growth promoting hormone, that is cytokinin. When you look into the word cytokinin, it is very interesting, something which is directly involved with the cells. So cell wall division is promoted mainly by the cytokinins. The first example, when someone asks you what is the example of a cytokinin, where do you find this abundantly, means the first example that will comes to our mind is the liquid endosperm of the coconut. Isn't it or not? Miller was the first person who has took out the cytokinin from the milk endosperm and named it as zeatin. After that, there was lot of work in cytokinins. So we find them cytokinins distributed vividly in both non-vascular as well as the vascular plants, and they are mainly found in the apical meristematic tissues developing seeds, roots, fruits. So they are present all over the plant parts. Okay, and we know that cytokinins are produced naturally, and there are certain kind of cytokinins which are artificially produced for commercial purposes. Okay, so the mainly wherever there are growing parts like newly setting flowers, production of new seeds, fruits, all these places are the ones where we find cytokinins in abundance. So if we are looking into the chemical nature of them, they are the derivatives of the purines, that is adenine. So it is 6-furfuryl aminopurine, and it is synthesized by 3 different pathways. Of course, the tRNA cytokinin synthesis is we find that the tDNA related CK synthesis can occur only in Agrobacterium. Others are found commonly in different kinds of plants. Okay. Now let us move on to the next one. Now let us try to observe the physiological effects of the this one, cytokinins. So, as I told you earlier also, they can trigger cell division and morphogenesis. So, we find that the cell division is promoted, and by proliferation of this cell division, morphogenesis, that is development of different plant parts, is possible because of the application or the presence of cytokinins in the plant. So, if the cell division has to occur, cell enlargement is the next one for morphogenesis. Not only that, so for breaking seed dormancy also. So, we find that in lettuce, dormancy is born upon it, which cannot be broken internally. So, application of cytokinins in this lettuce can lead to breakage of seed dormancy. Not only that, they auxins we know can cause apical dominance. These are counteracted by the cytokinins. Not only that, they also help in delaying the senescence. Senescence is what? Aging of the plant. This senescence can be delayed by retaining the chlorophyll and related proteins in the plant, in that particular plant area, by cytokinins. Do you know what is this is known as? That is retainment of the chlorophyll. It is called as Richmond-Lang effect. So, not only that, this particular cytokinins, when they are present in the plant, can confer resistance to the plant to different kinds of stress, abiotic stress that we are going to study about, or the attack of fungal pathogens or the microbial pathogens, as it may be. Not only that, they also have a triggering effect on the stomatal movement. Yes, and finally, they also help in enhancing the fruit quality, seed setting, development of the flowers and inflorescence in particular group of plants. So, these are the physiological effects of the cytokinins. Let us move further. You can see different pictures over here, where they have delaying senescence at one plant part and other ones. So, let us move on to the next one. Let us see the mode of action one more time. Now, they can very once again they have the similar effects what we have seen in gibberellins and auxins, that is, they have a direct effect on production of amino acids, metabolism of them, translation mechanisms. They do have connection with them also, and they also have this binding factors, that is, they are bound to certain specific proteins which can lead to further metabolic changes. Yes, and they can stimulate respiration also. It is a very interesting phenomenon that they promote respiration, and as we have discussed, they can retard or delay senescence by retaining the amount of chlorophyll and the related proteins in the particular plant. Now, the three examples that we have just studied, that is auxins, gibberellins as well as cytokinins, came under the category of growth promoting substances. Now, besides that, we have inhibitory substances also. The first one among that is abscisic acid. You might be asking me why we need abscisic acid, why we need inhibition. Imagine a situation where a plant keeps on growing continuously because of effect of only one kind of plant hormone. What could happen? Does its xylem and phloem support it? Does its root structure can support it? It is not there. There should be some limiting factor. There should be some counteraction. So, along with the growth promoting substances, we do also come across inhibiting substances so that the balance can be maintained. So, among the growth inhibiting substances, primarily we have to talk is the abscisic acid, commonly called as ABA. Now, this was discovered by Adicott, and it is a natural growth inhibiting plant hormone which is found everywhere in the plant. Okay. And not only ABA, we also have other phenolic compounds. I have told you before also, like cinnamic acid is there, ferulic acid is there. These are also phenolic compounds which can be act as a natural plant inhibitors. Okay. And besides ABA, we also come across artificial inhibitors also, like malic hydrazide is there, chlorocholine is there, these also, morphactin is there. These also are the commercial or synthetic plant inhibitors. So, we find that except in liverworts, that is certain group of bryophytes, we find abscisic acid in all plant groups, and in liverworts, we instead of ABA, we come across lunularic acid. Okay. So, if we are looking at the chemical structure, we find that it is a sesquiterpenoid and it is found in plus and minus enantiomers, that is they can rotate the light in right or left direction. So, the most active is 2-cis ABA is the most active naturally occurring what we known as a chemical agent in the plant. Now, let us move further. So, let us learn few more things about this popular natural growth inhibitor. So, it controls the uncontrolled growth of the cells, that is continuous enlargement, continuous growing is checked periodically. So, it acts as an natural internal check, and it remember that it is antioxidant, anti-gibberellin and anti-cytokinin. So, whatever effects they are going to show on the plant part, it is going to counterattack them. Okay. So, the movement of it is basipetal in nature, and from the leaves it gets transported to shoot tips and root tips, where through xylem and phloem, and where it can show its effect over there. Okay. So, let us go further now. Let us try to understand what are the various physiological aspects of this particular ABA. Now, if you are talking about ABA, the primary effect is growth inhibition, as we have talked about it. So, whatever counteractions of auxins are there, it is going to stop over them. So, it does not allow easy growing of all kinds of plant pods. So, it acts as a restrictor over there. Not only that, it also facilitates dormancy, that is in certain kind of woody plants we find that it induces bud initiation, so that the dormancy is maintained for few times. So, that the plant can be protected from the chilling effect or drying effect, whatever conditions, environmental conditions are there. Not only that, it promotes abscission also, that is that's what its name itself is abscisic acid. Yes. So, it promotes abscission of different plant parts, and not only that, it also has an effect on seed development and germination. And not only that, it also helps in promoting fruit growth and also flowering in few certain kinds of plants, not all. Actually it is an inhibitor in most of the cases, but in certain cases we also help in that very low concentrations of ABA can trigger fruit growth and flowering. And related to abscission is senescence. Senescence is old age, isn't it or not? So, it will trigger whichever plant part has attained certain amount of maturity or age, it helps in turning out that one and removal of that particular plant part through abscission. Not only that, the most important aspect, beside of course all of them are important, is stomatal movement. We know that stomata help in transpiration. Transpiration we know is a necessary evil. It is an important physiological activity which will actually trigger more absorption of water from the root. So, root pressure has to be created only when there is a transpiration occurring. But if it is an unending transpiration occurring through the stomata, it can also lead to dehydration, which is definitely not asked for. So, abscission, ABA when it is present near the God cells can actually lead to closing of stomata. So, it will act on that potassium hydrogen pump and closes the stomata. So, it is an anti-transparent, very effective natural anti-transparent is abscisic acid in the plant. So, natural check one more time. Okay. Not only that, in certain plants it also favors root geotropism, more movement of roots deeper into the soil. So, it favors root geotropism. Now, let us go further now. These pictures you can see how the effect of the stomata will lead to closing of the stomatal pores, and of course, the growth related processes you can see in the picture. Now, let us look into the activity, mode of action. Once again, it has a very bearing action, mode of action on enzyme synthesis and its regulation. Yes, and also movement of hydrogen and potassium ions, and related to the movement is the stomatal responses. And not only that, we find that they also inhibit at the translational level also. So, they act directly on RNA in one way or the other. And now, let us move on to the next one, that is the ethylene. Now, when we are talking about the ethylene, you might have observed that if we are placing a group of fruits in one place and leave them for a day or two, you find that even if there is one fruit, the other fruits also will become ripe. So, this ripening is brought about by this particular growth inhibitor called as the ethylene. Now, whatever growth substances till now we have talked, they are all different kinds of organic compounds. but ethylene is the only growth substance which is a gas. It is highly diffusible, it can very easily move into and out of the plants just like CO2, O2 or water. So, it is highly diffusible, its chemical nature also is very simple. So, CH2 bound by a double bond. Yes. So, we call ethylene as a gaseous phytohormone, and we find that ethylene is found in all plants and different part of the plant, but in very minute quantities, but we find that they are more characteristically seen in fruits, because the ripening of the fruit and further separation of the fruit from them is possible because of the ethylene over there. So, we find that in meristematic zone regions and in the nodal parts and in the fruit regions, usually find that ethylene is found in more concentration. Now, if you are talking about the biosynthetic pathway, there is no clear pathway how ethylene is produced inside the plant, but it requires methionine as one of the precursors. We know that methionine is coded by AUG, the first initiating codon, isn't it or not? Yes. So, not only that, now we find that auxins have a bearing on ethylene, that is, they regulate the production of ethylene. So, the auxins and ethylene production are correlated, and auxins can definitely affect the production of ethylene, and wherever auxins are present, ethylene is also present in those plant parts. So, let us go further now. Now, let us try to understand the physiological aspects of ethylene. So, since it is an inhibitor, once again you find the other part of these one, just like ABA, you find some common this one among their aspects. So, the abscission, ripening, effect on growth, degreening, senescence, flowering and dormancy are few physiological effects of the ethylene. Abscission we have already discussed, ripening is interrelated, as I told you. This ethylene gas, when it is produced in higher quantities, leads to ripening of the fruit parts. Now, this ripening can be exploited artificially also. You might be knowing when when the mango season comes, we find that they are the mangoes are harvested in the form of unset fruit, and later they are exposed to ethylene, and then the fruits are sent out for the market. So, it is one of the major horticulture practices. So, what does this do? This ethylene can lead to what is known as degreening of that particular fruit wall layer, leading to yellowing or maturation or ripening of the particular fruit. So, this particular ethylene can cause its effect on climatic fruits like bananas, tomatoes, but it does not have effect on non-climatic fruits like oranges or grapes. That is quite interesting over here. The second thing interrelated with the ripening is degreening. Now, what is this degreening? Degreening is a change of color, change of removal of the green color. What is the green color? The green color is because of the presence of the chloroplast. So, in the fruit wall, the degreening occurs because of exposure of that particular fruit to ethylene gas. So, ethylene gas along with light will lead to production of what is known as chlorophyllase enzyme. This chlorophyllase enzyme will digest the chlorophyll, leading to degreening of the fruits. Okay. Now, this methodology is used quite widely in the horticulture practices, and this is quite famously is known as a trickle method. Degreening is in commercial this one is known as the trickle method. Yes, not only that, in certain plants we also find that it also promotes flowering, and it can also help in breaking the dormancy in certain kind of plants. So, whatever chilling effects that are required or light effects that are required to break the bud dormancy can these ethylene can also cause in breaking them in absence of them particularly. Okay. So, you can see the ethylene action over here on different kinds of fruits, especially the bananas you can see over there. Yes, how degreening occurs and the ripening is occurring over here. Flower promotion you can see. Yes, growth you can see about them. Now, let us move to the next one. Let us see how what is their mode of action. So, they help in enzyme changes, they help in they have an effect on RNA synthesis, and RNA synthesis is directly dependent on DNA protein synthesis also, and the related this one physiological aspects. Now, let us look at the practical applications of the ethylene. Ethereal is the commercially used ethylene, which is artificially produced, synthetically produced and commercially used. Now, this is used popularly to accelerate the fruit ripening, so that the crop comes to handy and can be marketed and for the profit of the agriculturer. Yes, and not only that, it causes senescence in tobacco leaves. We know that the tobacco leaves are the commercially most important, isn't it or not? Yes, and they can also help in prolonging the storage life of the fruits and the related aspects. Now, let us move on to the next one, growth promoting. So, ethylene and abscisic acid, these were the growth inhibiting hormones. Now, we will go back to the growth promoting hormones, that is brassinosteroids. Now, this term is quite new because brassinosteroids are the newly understood, newly identified growth promoting hormones, which were first identified in Brassica napus, that is rapeseed, which is commercially oil producing seed. So, these brassinosteroids, after prolonged studies, has been identified in 44 different kind of plants, including 37 angiosperms, few gymnosperms, and in algae and pteridophytes also, and much more studies are going on them, and now the physiologists are considering them to be the 6th group of growth regulators. So, whatever growth activity that we have observed in auxins, gibberellins and cytokinins, the same kind of activity is exhibited by the brassinosteroids also. When you look into the word steroid, we associate this word with more of animals, hormones, isn't it or not? So, because their chemical structure is, it is a polyhydroxy steroid, that is why they are, and since they are found in Brassicaceae family members, they are aptly named as brassinosteroids, and we find that they are also produced in all plants of the part in very less quantities, very minimum quantities, but we find that they are maximum present in pollen grains and immature seeds. Okay, and the biosynthetic pathway through which these are produced is early C6 and late C6 oxidation process. So, along with other metabolic pathways, and it gives rise to various other intermediates along with the brassinosteroids. Now, look into the physiological aspects of the brassinosteroids. So, we find whatever we have studied in auxins, cytokinins and gibberellins, the same kind of aspects we can study over here in the brassinosteroids also, that is, they are growth promoting, they promote germination, they promote flowering, they inhibit senescence, and mainly the last point, that is, they help in stress tolerance to different abiotic factors. Now, we will just elaborate upon what kind of stress. Now, what do you think is a stress? Stress is an external pressure on a plant where the plant is in a non-ideal condition. So, it is not comfortable with its growth processes, it is not in a normal stage of growth. So, it is under stress. So, it cannot grow normally, nor its physiological aspects are growing on normally. So, it is under stress. So, what are the different kinds of abiotic stress? Stress related with temperature, stress related with either low temperatures or high temperatures, less amount of water, more amount of water, etc., etc. are the different kinds of stress what we see over here. These all different kinds of stress in different kinds of plants can be counteracted by the brassinosteroids. Okay. So, if you are coming at the mode of action, so they have an regulation we have on the gene activity. They can help in regulation of gene expression, related to that is the signal transduction, and also associated with plant growth, etc., etc. Okay. So, these are the different aspects of the plant hormones. Of course, 1 hour of time is not enough for us to cover all these aspects. So, just an overview of the plant hormones has been brought before you. Hope you have understood about them. And I now want to focus on few more aspects. As a physiology, as I told you in the beginning, is a very important part of PG entrance examination. I thought that why not I give you 1 or 2 tips on post graduation entrance also. Now, almost all of you are in either outgoing batch or the incoming batch is in final year, you might be definitely aiming for your PG at different universities, then how you are going to prepare for them. Now the mode of education has completely come to online, then why not prepare parallelly for your PG entrance also online. So books are definitely the best friends through which we can go for it, but online also there are so many different portals are there through which you can easily practice using mock test, and that too were free. So you can see some questions over here, yes, where you can keep on practicing and rating yourself about each topic. So for each topic you can type into the Google, search for that particular item, take an MCQ or a mock test. For example, I will show you this one. What is the treatment that is used for breaking bud dormancy is called? There are 4 options over here, and the right option is stratification. Yes. So another one also I would like to show you. This is how you can practice using the online platforms to prepare yourself for the PG entrances also. Now one question is there, which of these not a plant hormone? So there are options: nitric oxide is there, brasinosteroid is there, carcins are there, polyamines are there, jasmonic acid is there. Definitely among them carcins are the ones which are not the plant hormones, all others are the plant hormones. So this is from another web page where I have downloaded that screenshot for you, how you can practice. So the answers can be a single answer might be a right answer, sometimes more than one answer could also be right. For example, I would like to show over here the 5th question, which of the following is not a naturally occurring auxin over here. We have 4 options here. Here among that we find that 2,4-D is a synthetic one. We know that, isn't it or not? Yes. So even that one, and indole butyric acid, even that one is a synthetic auxin. So these are the type of questions you can come across on websites where you can continuous practice using one particular aspect like physiology. In physiology there are so many others, like plant hormones are there, respiration is there, photosynthesis is there. Each one you can type on that one. So I would like to emphasize on the different kinds of websites through which you can access these mock question papers or practice question papers. So you have TCY online website is there, which is a free practicing MCQs. You can practice. Easy Biology is there. Yes. Whizquiz is there. Yes. Not only that, NTA, National Testing Agency, also gives you a lot of practice sessions like this, where you can very easily freely practice and prepare for your any kind of upcoming competitive exams. So Toppers, you might be knowing, it is very famous. Yes, you can through Toppers also you can practice a lot of questions like this. So parallelly along with your semester theory, you can also prepare yourself very effectively using online portal, using various websites like that. Okay. Before I close off, I want to show different books, that is offline mode also, where the books that you can see in picture are quite good books written by experienced faculty, which can definitely help you in cracking your PG entrances over there. And among them is one of Telugu Academy books. I vouch for these Telugu Academy books because they are elaborately given in detail, and which would help the Telangana students specially to crack the PG entrances in Telangana and related universities. Okay. So these are all the different aspects that we have studied. Hope you have got at least a good amount of information regarding all of them. Okay. So I'll sign off. Thank you for patient listening. Stay home, stay safe, stay focused, and keep learning. Thank you then.